Welcome to The Explainer. Today, we're diving into a fascinating medical mystery featuring some tiny, furry protagonists. So, can observing adorable lab mice actually unlock the deep secrets behind our own eating habits? Let's find out. Okay, let's dive in. Binge eating means eating huge amounts without being physically hungry, feeling totally out of control. Meet our mice. We study them completely full, meaning any eating was driven purely by pleasure, not hunger. We gave them a choice: standard, boring chow, or an absolutely irresistible, high-fat, high-sugar sweet treat. First up, the baselines. One group got endless, 24/7 access to those treats, while the other got literally zero.
Now, what's really interesting, researchers created 4 distinct dietary schedules, mixing limited treat access with calorie restriction.
The test was super strict. Completely full mice were isolated and given exactly 2 hours with those yummy treats. The dramatic results? Simply restricting time with those treats triggered intense binge eating in every single experimental group.
Crazy enough, these little guys didn't eat less normal food to make up for those massive sugary binges.
Fascinatingly, combining calorie restriction with intermittent treat access caused the absolute fastest onset of binging in the female mice. Let's see how this builds. Researchers zoomed directly into the brain's reward center, analyzing neurotransmitter RNA to measure activity. They checked out 3 massive neurotransmitter systems to find the exact biological footprint of all this intense binging. Out of everything, the most crucial findings centered entirely around one specific gene, dopamine receptor 2, or DRD2. And this brilliantly illustrates how the binge groups uniquely and stubbornly maintained incredibly high DR2 expression. See, while constant snackers adapted and decreased expression, the binging mice completely failed to downregulate this crucial receptor. Here's the science. Higher D2D actually limits dopamine release, creating a surprisingly weak reward signal in their brains. Meaning, without that anticipated dopamine hit, the brain just craves more, directly driving that excessive, out-of-control eating. So, the crucial point is, these mice are binging simply to chase a missing dopamine high. Incredibly, these restricted access rodent models map absolutely perfectly onto the clinical symptoms of human binge eating disorder. This perfectly matches lisdexamfetamine, the only approved human medication which literally works by increasing that missing extracellular dopamine. These tiny heroes prove time-restricted diets trigger binges, highlighting the striatum's dopamine system as our core therapeutic target. If feeding schedules physically alter our dopamine receptors, what does this mean for diet culture? Thanks for watching.